So good afternoon, we can start. It is a pleasure to start this session. It's a session on, let's say, a kind of an unusual topic in epilepsy, but I think is building a lot of interest among clinicians and basic sciences as well, which is the topic of changes, alteration in myelin in focal epilepsies. So there is a lot of literature on that actually, which is hidden, because if you look at the literature on multiple sclerosis, you can realize that actually there is an association between seizures and demyelination in multiple sclerosis, when the demyelination affects mainly the cortical structure, not the white matter. And also, if you look at the data on animal models, you realize that indeed, almost all the animal models in which there is a focal lesion, there is also alteration of the myelin. So I tried to put together this session asking Tora Karadotir to give a first lecture on the general aspect, to remind us what myelin is, what the oligodendrocytes are, and how they are working to form a functional brain. And she has been working a lot. I put here one of the articles of her. It's a review article, very nice. I suggest everyone to read it. She is director of the Cambridge Center for Myelin Repair. She worked mainly on multiple sclerosis. She will tell us a lot about the normal myelin and also how that is altered and is regenerated after the damage in multiple sclerosis. Then we have an all-female session. There will be Laura Uva from Milano. She's a senior researcher, and she will tell you something about the finding that we have in human tissue of focal cortical dysplasia, which is part of what you can see, probably also in one of the posters. And during the COVID, we had time, we could not work with the hands. We tried to elaborate ideas and thoughts about the myelin. We wrote this review, an hypothesis actually, on how demyelination could affect excitability of the brain, and she will talk about that. Then Leonora Ronica, she is a professor of neuropathology at the University of Amsterdam. She will review the data available on human tissue, and she will give you an overview of the condition where alteration of the myelin have been demonstrated. And Erika Cecchini, she's giving the talk on MOGE, which is one of the emerging, let's say, one of the last identified specific focal cortical dysplasia where the alteration of the oligodendrocyte activity is prominent. Roland Koras was supposed to give this talk, and she will do an excellent job, I'm sure, substituting him. She's a researcher at the Erlangen neuropathology department in Germany. So I and my co-chair will be happy to lead this session on this new aspect, and I ask Tora to come to give his lecture. Please. I think that is. You have this.
Yeah, okay. So that should be good. Okay. Well, until my computer gets ready, I want to thank Marco and Christina for inviting me to come and give a talk here, come to Rome, and learn about epilepsy. So I will give, in my presentation, our perspective how neuronal activity can differentially regulate myelination. But before I start, I'm going to get us all to the same page. But before I get that, I have to do the declaration. I have no interest to declare, all free. So the human brain is equally divided into so-called gray matter. It's the computational part of a brain. That's where the neuronal cell body sits. And then it sends its actions through the white matter, and that's where information is being transmitted between brain regions. Segregating then a brain into these separate gray and white matter areas has been shown to be the best architecture to maximize neuronal connectivity with the minimum conduction delays, hence provide our maximum computational power. Yet neuroscience tends to have more focus on the gray matter, because that's where the neuron is. However, in the white matter is the oligodendrocytes that wrap their membrane around the axons in the central nervous system, producing this thick, compact myelin sheath. And this allows the propagation of the action potential to move much faster, makes it possible for us to move, think, listen to talks on epilepsy. However, in disease, the importance of myelin becomes quite clear when oligodendrocytes or myelin is damaged, as the action potential goes down slower or stops altogether, causing both mental and physical disability. This can happen in multiple conditions, but is best known, as said by Marco, in multiple sclerosis. What we also know from multiple sclerosis is that myelin can be regenerated. And when regenerated, it restores function. It gets regenerated because in our brain, throughout life, are cells called oligodendrocyte precursor cells, or OPCs, as I will call them from now on. They differentiate a multi-step process into fully myelinating oligodendrocytes. But how is the development of myelin occurring in the first place? This has been a question for a long time, and it was very beautifully mapped out by Paul Fletzig in the beginning of last century. Using myelin stain in post-mortem children, he finds around birth you have here dark blue is myelin. It's very poorly myelinated. And then really quickly in the first few months of life, we get quite fast myelination that continues into adulthood. Now, as we've just mentioned, lockdown, what do you do during lockdown? I gave my postdocs a task to read the whole literature on myelin from 1890 to 2020. And that's the old German literature, because I was lucky enough to have a German-speaking postdoc this time around. And we went over the histology from this period, matched it with the MRI data that is online free access, and new recent single-cell RNA sequencing data from human, for example, for databases like the Allen Institute. And what we found is this is a schematic diagram, is that in the beginning of human life, there's a great rate of change of myelin in the first few years. Then there's still myelination going on through childhood, but the rate of change is not as large. Then the second rate of change in myelination occurs during adolescence. Yet, myelin continues to increase throughout our lives up to a certain time. And now the time when this starts to change again varies dramatically between individuals. So it's very difficult to put exact date on it. But in our senior years, there's a third rate of change of myelin, is that it declines. Now the interesting correlation of these changes in myelination throughout lifetimes correlates with, for example, the first wave correlates with the neurodevelopmental disorders. The second one correlates with the onset of psychiatric disorders. And the third one correlates to onset of neurodegenerative disorders. I'm not saying that this correlation is causation, but there's a lot of things also happening in the brain at the same time. But what I'm highlighting here is perhaps myelin has an underlying cause to a range of CNS disorders that we haven't previously attributed. But how is myelination regulated in the first place? This is a question maybe that I've been battling with since I started my own laboratory. Our first experiments were to put OPCs in culture and plate them around with neuronal axons. What we found, even if we blocked all neuronal activity or glutamate receptors, the cells would differentiate and myelinate those axons. What we find is that this is, other people have found similar stuff, that they myelinate even fixed axons or nanofibers. However, when there is upregulation of growth factors surrounding the cells, growth factors known to upregulate synaptic plasticity, they become sensitive to neuronal activity. And this is particularly the time when they express the NMDA receptor. And then they differentiate only in an activity-dependent manner and myelinate active neurons. And we know that in young adults, if you reduce activity in the optic nerve or by chemogenetics, you get less myelination. Or if you increase the neuronal activity in young adults, you get more myelination. And we know that OPCs are present throughout the adult brain, both in humans and rodents. And these white dots indicate location of OPCs equally distributed to gray matter as well as the white matter. So our first question for this was when we'll see this is, are all OPCs the same? And this work was led by 3 PhD students and a postdoc, Helena Pivankova, that you may have heard earlier today, and just started her own lab in Prague and will become an expert in epilepsy, so keep an eye on her. Now back to OPCs. So we decided to record from oligodendrocyte precursor cells, or OPCs, from the first time they appear at the E13 in the animal throughout to the first year of life, when we start to see a decline in myelination, to see how they change their capacity to sense neuronal activity. When we record from the cells early on in E13, we identified that this fraction of cells, they don't express any ion channels or any receptors at all for neurotransmitters. Then they gradually acquire these channels by first channels appearing is voltage-gated potassium channels and K and AMPA receptors. Then you have voltage-gated sodium channels, and then you start to have the NMDA receptor with a peak of expression occurring at the peak of myelination and the loss of expression occurring when we start to see a decline in myelination. Lots of other experiments I'm pulling together now in a schematic diagram is that this is the conclusion that we think that there are cells that appear first as these naive cells with no ion channels at all, that then gradually acquire these channels, and that would be the migratory state. Then they turn into a highly proliferative state. They're all proliferating, but this one has a really fast proliferation, and that's the ones with voltage-gated sodium channels. Then they acquire NMDA receptors, and we think that marked the primed state for differentiation. And later they acquire so-called cell G1 arrested cell, the cell we think is arrested in arrested cell cycle state. And then they acquire what we putatively think is quiescent. Although I've demonstrated this as a line, it is not necessarily a timeline throughout the brain. We know that they start everywhere at birth, or not at birth, at E13, embryonic development, as naive cells. And then as the embryo develops and the animal gets born, the proportion of OPCs in different regions of the brain will have different cellular states. And these proportions will change with age and region until majority later in life start to have the quiescent phenotype. But there are some regions that will be different. So it's regional dependent and age dependent. Now, we have asked a lot whether these states are heterogeneity or cell states. And if it's cell state, it should be a fluid dynamic pathway between certain phenotypes. And in fact, what we found with a lot of experiments, that we can alter the states with neuronal activity or by giving drugs such as clementsine and metformin, which has been shown as the rejuvenating drugs, for example. But what is important for myelin regeneration, I believe, because in MS, which is one of the most common disabling neurological disease in young adults, occurs is that you have acute white matter lesions occurring here. This is a section of a person who had MS, and the dark blue is myelin staining, and the light blue is neurons. And there's a lesion where oligodendrocyte myelin is damaged. Now, this is in the adult, and we have OPCs, and they're going to be in different states. These OPCs, we know, can come in and migrate into a lesion, and we can see here's a lesion that has been regenerated. It takes up a little bit less dye because the myelin is thinner when it's regenerated. And that indicates that my OPCs have entered the lesion and differentiated and repaired and restored function. However, we also have a lesion here who is chronically demyelinated. Now, that indicates that is chronically demyelinated, has a risk of sustained disability, potentially because the OPCs have failed to differentiate. And the idea of OPC failure comes from the fact that when you stain these chronic lesions, you can see OPCs here in green and red axon around. However, if you talk to any of the neurologists, and I'm sure some of them are here in the room as well, is that you can have equal number of chronic lesions with a lots of OPCs in them, indicating failure differentiation. But you can also have equal number of chronic lesions with no OPCs in them, indicating what other people would argue is a failure of recruitment. So the argument in the MS field has been, why does remyelination fail in MS? So our question is, well, why does it? So to try to address this question, what we went for is a model where you just look at the model of myelin regeneration, not a model of MS, just how does the brain respond to a focal white matter lesion. And we went for the inferior olivar cerebral tract, where the inferior olive neurons send all the axons through the white matter tract called caudal cerebral peduncle. And this tract is that all the axons in that tract are myelinated. And when we inject the Ethereum bromide, we cause demyelination of these axons. Seen here, get fully demyelinated, but the neuronal cell bodies, because they're far, far away, are unaffected. So the neurons and the axons are healthy. And then this lesion gets fully regenerated. So it's a model of regeneration. We also know that in this model, once we have caused demyelination, it's very stereotypical, the events that happen afterwards. You get migration of OPCs, proliferation, then followed by proliferation of OPCs, followed by their different start of the differentiation, and then remyelination. And what we've come to talk about is that if we think about the OPCs as the states, I've talked about from embryo to the adult, what's happening in the lesion. So the question is, how is remyelination regulated? And this work is led by, has been worked by 3 postdocs in the lab. Helen Gautier started the work, and then Omar and Helena has been working on various aspects of this question in the last few years. So first is that we wanted to look at how does a neuron respond to a focal white matter lesion far, far, far down in the axon. We use genetically coded calcium sensor, GCAMP, in the inferior olive, and we implanted a photometer. We record activity in the inferior olive before lesioning and after lesioning. And here you can see a rat that has had a lesion and has the photometer. When we record before and even 3 days after lesion, there's no change in activity. But at 7 days post-lesion, there's a dip, a reduction in activity, but that upon remyelination is restored. So we have this transient depression of neuronal activity following demyelination in this tract. What we also do know that in the lesion, when we record from OPCs in the lesion, they receive synaptic input from demyelinated axons. And this has been validated in other white matter tracts, such as corpus callosum, by other people in other labs as well. And what we also know, that in the lesion, when we record from the OPCs, they receive and they sense and express glutamate receptors. But the onset of the NMDA receptor is about 7 or 8 days after the lesion. So it comes up later. Multiple experiments on these cells in the lesion identifies that the first cells appearing seems to be these naive cells I spoke about, the same one that we are at embryonic state, and then the migratory phenotype, the proliferating cell, followed by the primed OPC with the NMDA receptors. But now we also know that the activity declines at the time when the highest level of the proliferating cell types are present. So what is the role of this transient decline in neuronal activity? And this is very work taken up by Omar to look into that. So what we used is chemogenetics. This is the modified human muscarinic receptor. So the modified receptor that signals to GQ, if you're expressing that, you would depolarize the neuron, and that would increase spontaneous neuronal firing. If you express the modified receptor that signals to GI, you would hyperpolarize the neuron, and you would reduce spontaneous neuronal activity. Now, using this in a combination with the fact that if we use mini-pump to deliver drugs into the lesion, it kind of remains in the lesion. And at this time, we only have OPCs and macrophages and demyelinated axons in the lesion. So we can deliver drugs, but we also can alter the neuronal activity up in the inferior olive. So when we give clozapine or CNO, we will have activation of GQ or GI. When we do this by either delivering TTX into the lesion, blocking neuronal activity, we see an increase in proliferation. And if we do lower the spontaneous activity in the inferior olive neurons, we see an increase in proliferation in the lesion. But what we do see is if we keep the activity low throughout the regenerative period. Here's an electron microscope of the lesion. Green axons are remyelinated axons. Yellow, like colored, are demyelinated axons. And I hope you can see there are fewer remyelinated axons when you block neuronal activity in the lesion. Or if you lower activity of inferior olive neurons, you also get less remyelination. So we see that the blocking or reducing neural activity reduces remyelination. Even though it increased the proliferation of OPCs, it seems to have stopped them from passing on towards the primed state and differentiate. When we do the converse, if we keep the activity high, so we prevent the dip in activity after lesioning, we see a reduction in OPC proliferation. And if we keep it continually, just preventing that dip in activity, we see a less remyelination. However, if we augment or potentiate the rise in activity 10 days post-lesion, at the time when activity was starting to increase by itself, we get more remyelination. So to kind of take it together, normally once you have a lesion, the OPCs respond and start to proliferate, and the activity during this recruitment phase, the migration and proliferative phase, activity is down. Then during the differentiation phase, activity rises up again, and that can lead to proper remyelination. Now, if we changed activity during the recruitment period and kept it high, what we had a loss of OPC recruitment, and therefore a recruitment failure that led to remyelination failure. If we lower the activity, we got an increase in OPC numbers, but we got a block of differentiation. That would lead to remyelination failure. Whereas if we keep activity down and decreasing it at later time points in the activity phase, so not allowing activity to go up, we get a failure of remyelination. But if we increase activity at the differentiation phase when activity is increasing by itself and the cells are sensitive to neural activity, we get increase in remyelination. Now, I'm going to try to take this finding and mapping it on relevance of epilepsy, because activity and myelination is somehow linked. Now, depending on the brain region and the age of the animal, if activity changes when OPCs are in the naive to proliferating state, increasing neuroactivity of surrounding OPCs in this state will lead to hypomyelination. If you increase activity around when they're primed or in the cell cycle arrest, but still capable of sensing activity, you increase activity in this state, you will get a hypermyelination. It's a prediction. So I will end with that, and I thank you, and I thank my lab and my funders for helping us.
Thank you very much for this very interesting and, covering a lot of issues and topics. Other questions from the audience? Yeah. Erika?
Thank you very much for your great presentation. I was wondering, have you worked, or so if you worked with immunofluorescence techniques, were you able to identify, or did you identify a marker for actively remyelinating oligodendrocytes, whether it's possible to distinguish this subpopulation?
Of subpopulation of OPCs? Or
yes.
We are working really hard. So what we've done, and we worked with Helen on this, is to do PAT-seq in order to identify the transcriptomic signatures of each kind of a state. And from that, we are hoping that we get some markers that we can identify the different states and therefore do more global look in various tissues. So that's what we're working really hard on. We have putatively 2 markers for 2 states, but that's nowhere enough.
Okay, thank you.
One question at the end, down there.
Thank you for the talk. I was wondering if we know something about how excitability can control OPC functions. So are there signaling pathways or something, or hypotheses about that?
So I don't do a lot of intercellular signaling pathways, but many years ago, extracellular, yeah, well, yes. So what we find is that neuronal activity is a very good modulator to modify OPC states, so taking them out of G cell cycle arrest, for example, big synchronized activity can push them back into cell cycle to proliferate and then perhaps differentiate. We see that in certain states they will be capable of differentiating. We understand that if we feed animals metformin, now, you know what that does, I don't know, we don't know yet, but metformin seems to be able to alter the cell states more into the prime state. And, you know, that could be multiple mechanism. Where we know is other factors, such as BDNF and neuroregulin, also promote the prime state of OPCs if they're cultured around that alongside with signals from neurons. But apart from that, we haven't looked at what we predict is that microglia and astrocytes will have a big role in probably regulating the OPCs. But this is something that we haven't explored in detail.
Yeah, I have a question. You nicely demonstrated there is reciprocal interaction between demyelination and normal, let's say, activity. Is there any idea what might happen when the system is changing and is generating hypersynchronous activity or excessive activity? That means also big changes in potassium and so on. Is there any evidence that this can influence negatively or positively the remyelination or myelination?
So it's very difficult to predict. There's a lot of factors. I know from my very early work when we played with stroke, and the early stages of stroke, you can have a hyperactivity. OPCs are very sensitive to that. That potassium rises to very high levels surrounding the cells. So like the first minutes of stroke, there's increased extracellular potassium. OPCs will be sensitive to that. We identified back in the day that different types, so now I know different states of OPCs have different susceptibility. So depending on what state they're in, whether they're going to be susceptible. Other states were less susceptible. So I think we just have to start looking at it in a more nuanced way than saying OPCs as a one cell will be responding in this way or that way. It depends on what receptors the cells are expressing.
Yeah. No more questions, so we're going to follow. Thank you so much for the fantastic talk, and we're going to follow with our next speaker. So we have Laura Uwe, and the title is On Demyelinating Axons and Seizures.
Thank you. Good afternoon, everybody. Thank you, Marco. Thank you, Christina, for this opportunity. I have no disclosures. So myelin is constituted by the expansion of the oligodendrocytes. It plays several roles. For example, the presence of myelinated segment induces segregation of ion channels with sodium channels located in the node of Ranvier, whereas the potassium channel Kv1.1 are located in the juxtaparanodal region under the myelin. This segregation, together with an insulation property of myelin, enables the saltatory propagation of action potentials. Features of myelin segments, such as length, such as thickness, control the synaptic tuning, so the generation of brain oscillations and brain rhythm. Myelin also provides trophic support to the axon, and last but not least, it is involved in potassium buffering. Here we have human data about focal cortical dysplasia type 2B. It is a cortical malformation characterized by pharmacoresistance. It is highly epileptogenic, and it can be treated only with epilepsy surgery. Typically, it is associated, the typical radiological signs are an increase of the cortical thickness, blurring between the gray and white matter, and the so-called transmental sign. Histopathological analysis reveals the presence of demyelination. It can allow to distinguish from a perilesional cortex, which is normal, from the core of the lesion that shows demyelination, a disarrangement of the myelin, presence of dysmorphic neurons and balloon cells. Looking in the detail at the white matter, the hypersignal seen in the white matter typically correlated with a decrease of myelin content, which is confirmed also with the electron microscopy, showing a decreased number of myelinated fibers, a thinner myelin, dysmorphic cells, and edema. Analyzing the gray matter, when the myelin content was evaluated in the gray matter, no significant difference was observed between the core of the lesion and the perilesional cortex when the analysis was conducted on slices, same for the myelin basic protein. But when the analysis was conducted on electron microscopy, difference was observed in the lesion, where a decreased number of myelinated fibers was observed, and also the myelin was thinner, and interesting vacuoles was observed between the axon and the myelin. Here we have two groups of animal models. So in the first group, we have animals that present alteration of the myelin. And here we have the typical epilepsy animal model. And it's interesting to notice that there is a coexistence of two factors: alteration of the myelin and seizures. So this suggests that there can be a link between the two, and we hypothesize that the alteration of the potassium buffering can represent this link. Here we have a scheme proposed by Manichella and co-workers, in which they show the pathway which is followed by the potassium from the axon to blood vessels and pia. Briefly, the potassium which is released by the axon is uptaken by the KIR4.1 channels and connexin 29. It can travel across the myelin through gap junction composed by connexin 32. And from the oligodendrocytes, it can be transferred to the astrocytes through gap junction composed by connexin 47 and connexin 43, and then it can dispose into the blood vessel or at the level of the pia. And the authors propose that when a block occurs along this route, this can generate an accumulation of waters and ions, and in fact, they observe in this double knockout mouse from the connexin 47 and connexin 32 hypomyelination, seizures, and vacuoles. Here we have an example of a vacuoles between the axon and the myelin. And here there is another model, the KIR4.1 null mouse, that presents vacuoles in the outer aspect of the myelin. So findings suggest that the myelin status and between the myelin status and the seizure occurrence and the epileptogenesis, there is a link. And in particular, we hypothesize that when the myelin is compromised, also potassium buffering can be compromised. And in particular, we presented this hypothesis in which we expect that when there is a mild damage of the myelin, when an action potential is propagated in the axon, a release of potassium is induced in the extracellular space. But in this condition, we can expect that the potassium buffering still occur. But if this damage gets worse, we can expect that larger amount of potassium is released. At the beginning, it can induce just depolarization in a neighbor axon. Then it could, if this accumulation is larger, it can induce ectopic spikes in the neighbor axon. And it can, at the end, ignite a vicious circle that can sustain epileptogenesis and seizure occurrence. Part of this data and part of this hypothesis was suggested by the data that we obtained from experiment performed in the isolated guinea pig brain maintained in vitro. So I have to point out that the data I'm presenting are obtained from brains extracted from animals that are healthy. So we will compare different behavior of unmyelinated structure compared to myelinated structure. In particular, we recorded from the piriform cortex, which is part of the olfactory areas. So we recorded potassium shifts in 2 different positions simultaneously with field potential. And in particular, we compared the potassium shifts in the layer 1 of the piriform cortex that is a plexiform layer that lacks myelin, and in the cell layer of the piriform cortex that are cellular layers and present myelin fibers. In particular, we induced seizure-like events after administration of 4-aminopyridine, and we observed that at the seizure onset, there was a large increase of potassium in the layer 1 of the cortex, compared to the slower increase of potassium that we observed in the deeper layers of the cortex. We mimicked in a second group of experiment the pre-ictal activity, simulating the lateral olfactory tract, which is a bundle of fiber that projects directly to the piriform cortex. And in particular, we delivered trains at 30 Hertz, 5 trains at 30 Hertz, which is the frequency observed on top of this lower oscillation. And basically, we confirmed our results. So simulating the lateral olfactory tract by high-frequency stimulation, we observed that in the most superficial part of the cortex, there were larger and faster changes in potassium compared to the shift recorded in the deeper layers. Here we have the quantification of the potassium peaks we measured, and here connecting lines are connecting shifts that were simultaneously recorded. Then we look at the increase of potassium, the 2 recording positions, during the whole simulation. So we measure the potassium peak after each train. And you can notice that in the most superficial part, the increase followed a curve that can be described by a quadratic equation, whereas in the deeper part of the cortex, there is a linear increase. We found also differences in the decay in the 2 recording positions. So we measure the potassium at the end of the stimulation and 7 seconds after. And you can see here that the decay, so the recovery of the potassium after the stimulation is slower in the deeper part of the cortex. Sometimes the high-frequency stimulation induced activity after discharges, and in this case, we found that in the deeper part of the cortex, the potassium could even further increase, due, of course, to the spike activity. And finally, we interfere with the potassium buffering, applying barium chloride, which is a blocker of the KIR 4.1 channels. So the application of barium chloride induced larger shift of potassium and after discharges. So there was a larger increase of potassium together with a decrease of the threshold to induce after discharges. Between control condition and barium chloride, we maintain the same parameters of stimulation. So here we quantify the potassium shift before the application of barium in control condition and after barium, and you can see an increase. And also the decay was slower after barium chloride application. So our conclusions are the following. Potassium shifts are larger and faster in a non-myelinated structure represented by superficial piriform cortex compared to myelinated region represented by the deep piriform cortex, either during epileptiform activity or after high-frequency stimulation. We found that the dynamics between the two regions, monitor regions, are different in terms of potassium augmentations and potassium decay. So the dynamic of potassium was different in two regions that features different myelin content. But how to confirm that the damage of the myelin can induce an impairment in the potassium buffering? To prove that, we plan different types of experiments. In a first group of experiments, we will inject lysolecithin, which is a demyelinating agent, into one of the hippocampi of the guinea pigs. The animal will be monitored with the video EEG, and later the brain will be extracted for the immunohistochemistry and for the in vitro recording to monitor simultaneously the neuronal activity and the potassium shifts in control regions and demyelinated regions. We plan to induce demyelination also in organotypic slices, and they will be followed by the electrophysiological experiments as well. And then we would like to record from human slices obtained from the epilepsy surgery from patients that suffer from focal cortical dysplasia type 2B and temporal lobe epilepsy. And here are the people that I would like to thank that are helping me with this project. If you are interested in more details and more results about focal cortical dysplasia, please visit the poster presented by Laura Rossini. And whereas if you are interested in more detail about the comparison between myelinated structures and non-myelinated structure, please visit the poster presented by our PhD student, Gaia Bruno. Thank you.
Thank you very much, Laura, for a fantastic talk. So we open for questions. Sorry, I cannot see the end. Any question? Yeah, have two.
Thank you so much. I'm Mihai from Rennes, France. So which one do you think in focal cortical dysplasia is the chicken and the egg? Is it first the demyelination that causes epilepsy in the focal cortical dysplasia? Or are there immature neurons that don't produce myelin and don't encourage oligodendrocytes to produce myelin?
Well, I think that so far it's difficult to understand. We know that there are defects on the development, but what we can see is that there is, as far as I know, there is a defect in the production of, I mean, the hypomyelination is not accompanied by a degeneration of the axon. So what I think is that probably this defect in the formation of the myelin can cause a sufferance maybe in the axon, and this can induce a dysfunction in its property. But I think that we have to go on with the study to find the final answer to that.
There's a question here.
Hi, thank you. I'm curious. So you saw that you have thinner myelin, and you talk about less myelination overall. Do you know, have you quantified the number of axons myelinated? Is that changed? Or, because I'm just wondering, because thinner myelin in my books would mean myelin regeneration.
But you mean in focal cortical dysplasia? It's not my personal work, so what I know is that there was, so if you need more details, the Garbelli's group is the expert in our unit. But yes, there was a quantification of the myelinated axon with the density of the axon in the electron microscopy images, yes.
We didn't see unmyelinated axon because it's very difficult in the human tissue to recognize axon alone. Probably they are, in the majority, are myelinated axon. What we see was a decrease of myelin fibers and axon for consequence. Yeah, fewer.
Any other question? No? I would say we continue then. Thank you very much, Lara.
Now, please, it's the turn of Eleonora Ronica. She will tell us about the demyelination found in different forms of focal epilepsy.
Thank you, Marco. It's really a great pleasure to participate to this session and this interesting topic. So my task is to address myelin pathology, also beyond the demyelination, throughout a different type of focal epilepsy. We will see from acquired to genetic epilepsy, going from animal model to the human pathology. These are my disclosures. So what I will do is, I want to start defining myelin pathology in the context of epilepsy, particularly in the context of the process called epileptogenesis, defining what is hypodemyelination, dysmyelination, myelin plasticity, and introducing the concept of adaptive versus maladaptive myelination. And then I will provide some example of myelin pathology in both acquired genetic focal epilepsy, so going from model to the human pathology, but also back to the model, because this will help us to understand the biological convergence which connects myelin pathology with epilepsy, so seizure development, progression, and the comorbidity, which are all part of the process called epileptogenesis. So as we know, that is a complex process, chronic, dynamic, which is the results of different mechanisms, which all together contribute to the development of a pathological network, because epilepsy is a brain network disorder. So our challenge is to dissect the different components at the level of DNA, RNA, the protein, at the cellular level, their interaction, the impact of the network, and here we focus on the myelin pathology. So what I did, I went back to the literature, look at the long period of time, and what I can say, as I could expect, the first publications are really referring to the classical genetic epilepsy, which affected the white matter, where there is clearly a problem in the process of myelination. But the more recent publication actually provide information that there is a myelin pathology occurring also in focal structure epilepsy from acquired to genetic. And what you see also in the time that change also our view to interpret white matter alteration, in general myelin pathology. It's not only the pathology of the oligodendrocytes, but actually you have to consider the oligodendrocytes as part of a network interacting with other type of cells. And these are neurons, first of all, but also glial cells as astrocytes, microglia, and also the interaction with blood vessels. So when, as a neuropathology, we refer to myelin pathology, we can describe hypomyelination, so we have insufficient myelin deposition. We can describe dysmyelination, so abnormal myelin deposition, and the demyelination, so we have loss of myelin or destruction of myelin. Actually, in epilepsy condition, we can have a combination of all these three pathologies. You can imagine if you have a genetic epilepsy, which affects the maturation of OPS, the oligodendrocyte progenitor cells, you can have a problem in the formation, you can have insufficient myelin deposition, but also abnormal myelin deposition. And then you have the seizures, so you have activation of all the process. So for example, we investigate inflammation, oxidative stress, which later on may also compromise, also in other regions, the myelin with then destruction of myelin or interfering with regeneration. So to understand myelin pathology in the context of epilepsy, I would like to recall what has been already introduced in the first presentation, the myelin plasticity. And I would like to focus your attention on the cortical myelination, which is, as we just learned, is a lifelong process. So the new oligodendrocytes are formed continuously and can be integrated. And this integration in the network is depending, first of all, and not only, from neuronal activity. As we just hear, based on experimental model, we know that there is an activity-dependent myelination which continuously, throughout all our life, regulate, fine-tune the neuronal network dynamics. And we learn from experimental model, using model which modulate neuronal activity in different ways, optogenetic, hemogenetic, spatial learning, memory, motor learning, sensory deprivation of sensory enrichment. So this brings me to the term, a definition of the adaptive myelination, which has been introduced recently really to describe this physiological process, this activity-regulated myelination, which occurs during development in relation with motor function, learning, memory, sensory function. And this is based on the concept that there is a continuous crosstalk between the oligodendrocytes, actually the precursor oligodendrocyte, precursor cells, and the neurons. And we just learned that this contact can occur at the level of the soma, of the axon termina, and is also depending on the developmental changes and the status of these OPS, which do express indeed initially potassium channel, and later on also the sodium channel play a role, then we have the AMPA receptor, then we have the NMDA receptor. The GABA play also an important role during development, regulating the function and the interaction between neurons and OPS. So one example of adaptive myelination, which I find particularly interesting, refers to the GABAergic interneurons. For example, it's been shown that this interneuron subclass, really subpopulation, have different profile of myelination. So this crosstalk is really specific for specific population of cells. So if you have an alteration of this communication, it occurs during development, then you have an impact on the network with an impact on comorbidity, so you can have, for example, here a problem with cognitive behavior. This, I think, is important for us to understand the consequence of a myelin pathology in the context of epilepsy for the comorbidities. So then has been also introduced this term of maladaptive myelination to describe the consequence of abnormal neuronal activity, which interfere with this physiological process, shaping the network in another way, with consequences which contribute to dysfunction. And this is a nice recent paper which described, for example, maladaptive myelination in this model of actually generalized epilepsy with absent seizures. And they show here that there is actually an increased oligodendrogenesis within the network. And they show that in this model occurs after seizure development, but they also show that contribute to epilepsy progression. But here we are dealing with generalized epilepsy. What about focal epilepsy, which is the topic of today? So I will show some examples starting from models of acquired epilepsy in the past, throughout the years. Different models have been introduced, used, and white matter pathology and myelin change have been reported in cyanuric acid, pentylene tetrazole, tetanotoxin, electrical kindling, lithium pilocarpine. And we know that we have also alteration of myelin in the model of traumatic brain injury. Okay, we have alteration of myelin. The first question, and it's one of the questions that answer Laura with her work, is what is the effect of altered or reduced myelin content? We saw already in the previous presentation. These are old studies which actually with different approach show, for example, interfering with the synthesis of myelin or using a model of toxic demyelination. They show that there is a relationship between loss of myelin and increased susceptibility to seizures. The second question, which was also addressed in the previous presentation, is, okay, is all the consequence of seizure? Or you can find white matter alterations of myelin pathology prior to seizure. Also here, we can say yes. Now, this is just example from old study. We show that we can have white matter alteration in the spontaneous epileptic rat model. And there is also evidence that if you have a delay of demyelination and neurodevelopment, you can have explain increased susceptibility in the kindling epileptogenesis. So this brings me to the genetic epilepsy. Do we expect myelin pathology in genetic epilepsy? What we know, what we learn from models? For sure, we learn a lot from model of mTORopathies. This is not a surprise. I will come back to this because mTOR is important. It's one of the key pathways for brain development and function, and it's not a surprise. Regulate also the development, differentiation, and maturation of OPS. But in model of tuberous sclerosis complex has been also introduced another mechanism. I'm referring to this work. We show that alteration in neuron function may affect the myelin. So we can talk about non-cell autonomous effect involving, for example, growth factors. What about other genetic epilepsy? Do we expect myelin pathology? And also here, experimental study may help to understand the function and the role during development of, for example, ion channels, which are target of mutation in developmental epileptic encephalopathy, talking about the sodium channel, potassium channel. These are really essential for the normal development and differentiation of OPS and the process of myelination. And also the neurotransmitters, GABA. GABA is really important in the process of myelination. So if you have any alteration of the GABAergic transmission affecting GABA A or GABA B receptor, you could expect an impact on myelination. Not only GABA, we just hear about the key role during development of a receptor as an NMDA. And NMDA really, once that they are activated through also growth factors, BDNF, are really key, play a key role in switching OPS to this activity-dependent myelination mode. So if you have an alteration of this pathway, we will have an impact on myelination. And I would like to mention also these genes, CDKL5, that you know as a target of mutation in developmental epileptic encephalopathy. Also, this specific genes is involved, in this case, in the adult OPS maturation and myelin repair. So if you say, do you expect myelin pathology in genetic epilepsy? Yes. So now we go to the human pathology. I'm neuropathology, so how human tissue can be used and has been used to add an additional information. And we start from acquired epilepsy, and these are studies we show using material obtained autopsy or surgery that indeed in temporal lobe epilepsy, you have some degeneration of myelin. You have some reduction of myelin basic protein or other related protein. You can have also even a proliferation of these oligodendrocytes, which has been reported in TLE, a little bit similar beyond astrogliosis. And for sure, we have alteration of myelin if we look at tissue from patients with traumatic brain injury, because white matter injury is a component of TBI. Mild form of TBI, they also cause a complex pathology, which can be associated with not only reduced but aberrant myelin synthesis. And I would like to highlight this study, which are really interesting because it combines imaging, also in tissue, ex vivo imaging, and the clinical pathological correlation. Particularly, I would like to highlight this study of my colleagues, Rita Garbelli and the whole team, which was several years ago, but it's an important study because using ex vivo MRI and also looking at the neuropathology, electron microscope is going at the EM level, show and dissect the mechanism, the pathology underlying the blurring in patients with temporal lobe epilepsy, showing that there is axonal degeneration, there is a reduction of number of axons. We can speculate about other mechanisms, for example, the role of age accelerate pathology of small vessel and the white matter, which required additional information. And all these studies show that the white matter alteration have an impact also on the comorbidity. I would like also to highlight this important study, an imaging study, because it refers to a large cohort. It shows that white matter abnormality are really a commonality throughout the different epilepsy syndrome, in this case in adults. And this extends also to tumor-associated epilepsy. Indeed, in low-grade epilepsy-associated tumor, think ganglioma, DNT, there is a rarefaction of the white matter, which has been correlated with loss of myelin and axons. And now, as if you see in the literature, there is an increased number of studies which focus on the impact of tumor and tumor-associated epilepsy on the subcortical brain structures, which affect also the connectivity and will involve also the white matter and may explain also the comorbidities. And you can use also human tissue to analyze the molecular network. You can use proteomics, transcriptomics. In this study, we use transcriptomics, combine temporal lobe epilepsy with hippocampal sclerosis and enteropathies. And these molecular studies show that indeed there are some commonality involving dysfunction, not only of neurons, astrocytes, microglia, but also oligodendrocytes. But it's also clear that the process of myelination is specifically impacted in enteropathies. Is this a surprise? Not really, because mTOR, as I mentioned before, is a key pathway for brain development and function, regulate growth, proliferation, migration, and also the maturation of oligodendrocytes. So any alteration of this pathway, which can be genetic or acquired, particularly if it's early during development, has impact on myelination, so may interfere for proper myelination. And clearly, alteration of myelin have been already reported from the first description of the cortical lesions, so-called tubers in tuberous sclerosis complex. And this is a brain, autopsy brain. You see the tuber increase intensity and the ex vivo MRI, which correspond, as expected, to a loss of myelin, here depicted with luxofast blue simple staining. I would like to highlight all the more recent study, imaging study, which really provide evidence that this early white matter development is affected in TSC, and actually is more affected in patients, in children with comorbidity, including autism spectrum disorder, really provide evidence that the pathology of myelin and the white matter alterations, so in general, are important to explain comorbidities. And a basic neuropathology study confirmed in TSC that there is a loss of myelin, cortical tuber, may be different from tuber to tuber, and he is completely gone. And these are indeed the tuber C, these tubers from children with severe epilepsy, early seizure development, and also severe comorbidities. But also we learn that myelin pathology in a cortical tuber extend beyond white matter. There is a decrease of number of OLIG2 positive cells, not only in the white, but also in the gray matter. These intrinsic, in our case, correlate also with the presence of inflammatory cells as T cells. But more important to acknowledge that the number of oligodendroglia and the myelin-specific protein correlate with cognitive performance of our patients, and even correlate with the presence of important comorbidity for life of these patients as autism spectrum disorders. And it's only TSC, not. We just hear from the more recent study of Laura, that is also FCD type 2. These are really the first study which have been discussed hypomyelination. At that time, so before the study of Laura, the discussion was, is a reduction of myelinate axon or something which impair the maturation of oligodendrocytes? And this neuropathological study using marker which allow to follow the maturation of oligodendrocytes from OPS to mature oligodendrocytes indeed show that there is an impaired turnover of these cells. In some region we can have hyperplasia, but these cells that do not reach the mature stage which allow to produce normal and sufficient myelin. And this is not only true for TSC and FCD type 2, but this is a study also recent which showed that it's also in FCD2A, so in all the enteropathies. This study showed that there is in cortical layer 5, there is something wrong of thinner myelin, and they show that there is also a problem with the maturation of oligodendrocytes and the proliferative capacity. Molecular study combining human tissue with experimental model FCD also confirmed that there is a problem with oligodendrocytes and the process of myelination. And this is an in vitro study which was useful to understand, really to provide evidence that there is an intrinsic failure of the oligodendrocytes in enteropathies. In this case, they used the nanofiber cultures of patients derived primary oligodendrocytes, TSC2 knockout cells. They show that indeed there is a reduced oligodendrocyte turnover, which is related to the decline of myelin. But more important, they show that it's mTOR dependent. So if you use everolimus or mTOR inhibitors, you can restore the myelin formation, and you can normalize this proliferative activity of the cells. So what we can say related to enteropathies, that myelin, and we know, is affected in enteropathy. The myelin pathology is really part of this complex network of mechanisms which contribute to seizure development, progression, comorbidity, but it's not isolated. Myelin pathology has an impact, and we just hear network imbalance. Network imbalance contribute to myelin pathology. But we also found evidence of a crosstalk between other type of cells. We talk about T cells, but for sure also astrocytes and microglia. So inflammation is also associated with myelin pathology. More important, we have experimental and clinical evidence that myelin pathology is associated with cognitive comorbidity. But we are dealing only with enteropathies, not. And to make a link with the next presentation, I just mentioned MOGA, so we will hear more from Erika with this MOGA, difficult name, but one part of the name is oligodendrocyte hyperplasia, so something wrong with oligodendrocytes. There are some patch area of hypomyelination. And here we have another challenge we will hear in the next presentation, how we can link, in this case, N-glycosylation with hypomyelination, and how this may contribute to the phenotype of these patients. So I hope that I convince you that myelin pathology is a hallmark of epilepsy, which is really a key component of the process of epileptogenesis from acquired to genetic epilepsy, so not only enteropathies. This knowledge is important because it offers opportunity, challenge, particularly in our effort to modify the course of the disease, impacting not only the seizures, but also the comorbidities. And with this, I would like to acknowledge the support, particularly the national and international collaborators, and thank you for your attention.
Thank you, Eleonora. Are there questions for this, please?
So, thank you. What effects can antiepileptic drugs have on OPC? So, like progression or maturation?
That is an important question because when we analyze transcriptomic data, we have always to consider the impact of anti-seizure medication, which have been used for sure for a long period, and in TSC since the first months of life when seizures appears. We didn't see, for example, in analyzing our transcriptomic data, any correlation with specific medication. But for sure, you have to consider this, and there are some drugs which I know that have an impact on myelination that we have to consider in the evaluation also of the alteration that you can identify on MRI. So it's something that we have to consider in analyzing the data, and that can be also studied for specific drugs in vitro models. Okay, thank you.
Time, unfortunately, we have to continue. So our last speaker of the session is Erika Cecchini with altered oligodendrocyte function and mild cortical malformation associated with epilepsy. Thank you, Erika.
Okay. Yeah, okay. Thank you very much for the nice introduction, and I declare that I have nothing to disclose. So I want to start with giving you a brief introduction or a refresh on the international classification of focal cortical dysplasia. So historically, there has been much talk about neurons and gray matter when referring to epilepsy, especially when referring to epilepsy associated to focal cortical dysplasia. Or FCDs, and this is not the same for white matter and oligodendrocytes. As a matter of fact, until 2022, the international classification of FCD was strictly descriptive of lesions of the brain related to the gray matter. But it is in 2022 that the ILAI task force decided to provide an update of the FCD classification to include the new knowledge acquired in the field of FCDs. And it is here that they decide to include a new category, so the category of white matter lesions, and within this category we find MMCDs and MOGE, which stands for mild malformation of cortical development with oligodendroglial hyperplasia in epilepsy. And today my talk will focus on this pathology which distinguishes itself exactly for the oligodendrocytes. So, MOGE is a relatively new, it's a new disease which is associated to drug-resistant epilepsy and which is found mostly in the pediatric population with a location prevalent within the frontal lobe. So, the best way for me to talk about you about about MOGE is by its histopathology. So, the first thing that should pop into your eyes when you look into this hematoxylin eosin staining of a brain tissue section from a MOGE patient is the increased number of oligodendrocytes, so the cells of the white matter. And this is also the first features that allows you, as a neuropathology, to distinguish this, to get a diagnosis of this condition. And the second feature is the presence of heterotopic neurons within the white matter, which I have highlighted here with black stars. So on this right panel, you can see distinctively oligodendrocytes and heterotopic neurons in a control tissue and MOGE tissue. So the new knowledge acquired also into the clinical imaging field allowed clinicians to distinguish two main subtypes of signals in MRI for MOGE patients. So we can see a subtype 1 in young children and a subtype 2 in older children, or let's say children over the age of 5. So what you can see here, the characteristic of the subtype 1 is that you can see this hyperintensity at the level of cortical medullary junction, and this is possible to see by FLAIR or T2-weighted MRI. The second type, the subtype 2, is characterized by this reduced differentiation of the cortical medullary junction. And here you can see that it's hard to distinguish between gray matter and white matter, and this is probably due because of the increased signal of the adjacent white matter in the cortex. And what is interesting is that this observation at the level of MRI are matching the histopathology of brain tissue samples in MOGE patients. So what you are seeing here is a staining with Nisl LFP, Luxol Fast Blue, which is a stain that we use to visualize myelin into MOGE brain tissue. And you can clearly see this patchy pattern that is present in the tissue, which is highlighting a condition of hypomyelination. You can clearly see the difference between how a, let's say, properly myelinated brain is supposed to look like, because you can see that this color of the LFB looks olfactory is very homogeneous. So these results have not been published yet, but our group in Erlangen at the Neuropathology Institute, in the attempt to visualize the distribution of oligodendrocyte within the Moge lesion, we created actually heat maps of oligodendrocytes. So these heat maps indicate the density of the oligodendrocyte within the lesion, within the tissue, within the lesion. The area in blue or green represent low density of oligodendrocyte, and the red area indicate a high density of oligodendrocyte. And what is interesting is that this increase of the oligodendrocyte is exactly matching with the areas of hypomyelination. So one theory, one hypothesis is that because of this huge crowd of oligodendrocytes, there is somehow no room left for the instatement of myelin. Another, on the contrary, another hypothesis is that the tissue, in the attempt to compensate for hypomyelination, is recruited more oligodendrocyte to provide for new myelination. So, in the attempt to explain this histopathological phenotype, some groups, like the group from Stephanie Bullock, performed deep targeted gene sequences on Moge brain tissue, which was collected from multicenter cohorts from pediatric individuals, and they performed laser microdissection on the tissue to analyze cell population by cell population. And what we discovered is that oligodendrocytes and heterotopic neurons carry pathogenic variants within somatic pathogenic variants within SLC35A2 gene. Now, our group in Erlangen, we went through the archives of the Neuropathology Institute to collect a cohort. So we were able to include in this cohort 83 individuals with Moge, which were genotyped for SLC35A2. And what we actually see, what we saw is that 41 individuals out of 83 carried a somatic pathogenic variant within the SLC35A2 genes. This means that about 50% of tissue samples with Moge carry brain somatic variants in SLC35A2. So this gene encodes for a UDP galactose transporter. This is a transporter that moves galactose from the cell cytosol into the Golgi lumen. Now, galactose is a molecule needed for post-translational modification of proteins and lipids. It means that this molecule is necessary for the proper function of the protein, of proteins and lipids, but also for molecular trafficking, in sense that it tells, determines the final destination of the proteins and lipids. In particular, we are interested in lipids because there's a class that is strictly related to proper myelin function and formation. Now, what we detected is, so what is being recorded right now is that the most frequent variant in SLC35A2 protein is our frame shift insertion of deletion. This determines the production of a truncating variant of the proteins. This means that a loss of function of the gene determines a reduced expression on the protein. And this has an effect on myelination. We wanted to investigate this at the ultrastructural level. So what you can see here on the left column is a control sample at different magnification. On the right, you can see Moge within the lesion core of Moge. And what we could actually detect was a drastic reduction of multi-layered of axon with multi-layered myelin sheath. At a higher magnification, you can also see the presence of a higher number of oligodendrocyte with single-layer myelin sheath. But also we observed this very interesting, this kind of myelin conformation around this axon. So one possibility is that this myelin represent, so what is this myelin doing around the axon is an early stage of myelination, meaning that the myelin is about to wrap up the axon. Another theory is that we are assisting to a myelin outfolding in terms of that the myelin sheet is not able to stay anchored at the neuron, and this is called this detachment of the myelin sheet from the axon. And we actually were able to quantify these results. So in this graph, very simply, we quantify the myelin content within MOGE core lesion and in control tissue samples, and we actually saw a significant reduction of myelin content within the core of MOGE lesions. Now, I want to conclude this talk with talking about the most recent pilot trial of precision medicine, or at least one of the most recent. So in this pilot, considering from past publication where patient with congenital disorder of glycosylation due to SLC35A2 mutation, in this patient was detected an improvement of the symptoms following administration of the galactose. So the authors of this paper tried this approach even for MOGE patients with SLC35A2 variants. And what we could see after 6 months of the galactose administration, that after 1 month, 1 patient out of 6 was free of seizure, and 2 patients achieved a reduction of frequency seizure under 50%, so a seizure reduction over the 50%. So the take-home message I want to take with you about MOGE today is that MOGE is a condition which is characterized by increased cluster of oligodendrocyte and heterotopic neurons within the white matter. It is diagnosed by first these 2 features, but also by the presence of patchy areas of hypomyelination, and I'm addressing this especially to the clinicians present in this room. About 50% of the tissue samples carry brain somatic variants within the SLC35A2 genes, and that patient with MOGE, SLC35A2 variant following the galactose administration actually had an improvement in their symptoms. I want to thank all my colleagues and our collaborators, and I want to thank you for your attention.
That's very good. Thank you very much, Eric. A fantastic talk. So we open for questions.
Thank you, Erika. Very nice. My question is, did you have any direct evidence that in this patient with mutation, for example, in comparison with patient without mutation, there is a real defect in glycosylation in the tissue brain, with other technique, for example, not with histology?
Actually, no, we couldn't. Do you mean whether we saw differences between MOGE with or without mutation in terms of myelin? It's actually
in terms of glycosylation, pattern of glycosylation, lipid glycosylation in the tissue brain?
That's something.
Or is something we are interested in?
This is a theory. It's a theory. Because from the literature, so the most probable class of lipids that is involved in myelination is phingolipids, we saw this relevance of this class of lipids into the myelination, but this is rather hypothesis based on literature than something that we could actually identify in lab.
Any other question? Yeah, at the end.
Yeah, thank you for the wonderful talk. So I have a slightly, a corollary question, like with MOG-associated demyelination coming up in a big way in the last, decade or so, particularly in the pediatric age group. Mm-hmm. And also the spectrum is quite variable. And amongst the demyelination entities, it is the one which has maximum reports of associated epilepsy. And your mutations are there only in 50%. So your thoughts on, what proportion could be actually an underlying MOGAD and role of immunotherapy in these type of cases?
Okay, I think I'm not prepared for this kind of clinical question.
Then I'm really
because
sorry.
Actually, I didn't get to disclose. I'm a neurobiologist, so okay. I'm not really into touch with clinical, with therapists and patients.
Okay.
But if you want, I can provide you contact of the neuropathologist in my laboratory, and you can maybe discuss together this topic.
Okay. Thank you so much.
Thank you. Sorry. Thank you.
More questions? No? There.
Yep.
I pose a short question for maybe Eleonora is better to respond to it, or any member of the people that presented the wonderful presentation. So if we have this theory that epilepsy is also a disease of white matter, why don't you see in clinical pattern, like the patients with really white matter disease, multiple sclerosis, etc., vasculitis, more epilepsy, because they have an increased risk, but this risk is not enormous. So in our cohort of 600 SEEGs, we only had 3 pharmacoresistant multiple sclerosis patients that needed an operation. It was not linked to their multiple sclerosis lesions, was mostly temporal epilepsies. So I don't know if you have any hypothesis at this moment, or...
Can you just bring the microphone,
yeah,
please?
Sure.
Yeah.
Yeah, for MS, you can also answer about that. Normally what we see, we have patients which they have seizures, and they are most associated with lesions in the gray matter or involving the hippocampus. So that is, but clearly, as I mentioned before, the white matter pathology has different, the mechanisms which contribute to the myelin pathology are different in different type of epilepsy. You have to consider the time when the injury occurs, can be genetic or acquired injury, because as in the first presentation we saw how important is the timing, that you can have different effect of a neuronal activity or hyperactivity depending if we are dealing with OPS at the early stage or the OPS when they are at the later stage of maturation. So it is more complicated, but in MS is reported and recently is also described in a higher number of patients correlated with the gray matter lesions. I don't know if you want to add something, because it's your topic of...
Well, it's not really my topic. I'm a neurobiologist. But what I want to add on to is, I think in MS you have very focal white matter lesion. You don't get very diffuse white matter lesions, as we've just been seeing here. And I'm just going to add, because I've been thinking about it right now, every time if I do a transgenic mouse model where I want to fiddle around with myelination or OPC differentiation, and if the animal house rings and says we have seizures, we are on a winner. We've stopped myelination. So I don't know how that links, but in the myelin field, if you prevent myelination development, animals will seizure. And that's our first indication that we've done things right. Oh, well, wrong.
For the MOGA, it will be really interesting to look in these two groups. First, the group where you don't find a mutation, maybe we don't find a mutation because it's a low level, so that's okay, we can exclude. I think the suggestion of you to look at the glycosylation, you can do the technique. I think the group in Korea, they did this approach to look at the N-glycosylation activity. So you have to have fresh material, you cannot do on paraffin. It's more challenging technically.
Yes, I also want to add that this is a spectrum of pathology, because when we talk about this mutation, we have a variant allele frequency that until now we saw ranges from 2% to 52%. So it's also very crucial in identifying the lesion core, and it's like trying a bullseye to hit the right spot to identify the mutation.
Yeah, we have a question.
Thank you, Erika. I think yeah, one more question.
Yeah, one more question. And then unfortunately we have to wrap up.
Thank you very much as well for the lovely presentation. I would like to play a little bit of devil's advocate. So I'm not sure if I haven't missed it in the presentation, but you mentioned that you only detected the SLC35A2 variant in 1/2 of your cohort.
Yes.
So my question would be, how do you explain it? Do you think that MOGA is caused by another gene, or was it a methodology issue, or can you be sure it was MOGA?
So this is something I'm talking about tomorrow on the remnant 50% of the cases that we were not able to identify with to find a mutation within the SOC35B2 gene, so X-related. I can give you a brief spoiler that we think it might be Y-related, Y chromosome related. But as I said, could also be that I think the complexity is also to find a proper spot, but also sometimes to have the proper, sensitive enough instrument tool to identify the mutation, because also identify 2% variant allele frequency is pretty challenging because we're talking about a mosaicism, so it being somatic, it's not present in every cell, so that's the most challenging part, I would say.
Yep. Yeah, I think we have to close. I see people coming in for the next question, the next session. So I think that it is clear there is a correlation between changes in myelin and epilepsy. Whether there is a correlation or a causative role in myelin alteration and epilepsy, this is something that we have to discover. So we open up more questions and responses. Another thing to think about it is that alteration in myelin can be, the pathomechanism of alteration in myelin can be different in different diseases associated with epilepsy. So in MOGE will be different from the post-traumatic loss of myelin. And the other thing that has to be found out is whether there is a causal correlation between the changes in myelin content and seizure activity, so the functional aspect of epilepsy. So this opened up the work for the next couple of centuries, and we will do it.
Thank you very much, everyone. Thank you very much to all the speakers.